Hello children, in the last lecture we have been discussing regarding the plasmodesmata. So similar structures, the plasmodesmata are present generally in the plants, and the similar structures I have told you that cell junctions are seen in the case of animals. But we are not discussing with the cell junctions here, we will be discussing this part in the zoology. Okay, after this we will be discussing with the next structure, that is plasma membrane. Okay, let's begin. So this is plasma membrane, children. Plasma membrane encloses each and every type of cell, eukaryotic, prokaryotic, or any type of cell. If you take the case of the plant, animal, bacterial cell, all the cells will be enclosed by this plasma membrane. So it is a protective cover over all the cells, prokaryotic, eukaryotic, animal, plant, bacterial cell. All these organisms and the cells, they will be made up of this plasma membrane. They will be covered by this plasma membrane. Not only the cells, children, the cell cytoplasm will contain many organelles. All these organelles in turn are made up of the plasma membrane, and hence we say that the plasma membrane is universal in nature. The structure is universal. Why? Because the same structure will be present. If you take the case of a bacterial cell, animal cell, or a plant cell, the structure will be same. It is uniform. And not only that, it is present without exception. It is seen in all type of cells, and hence we say that the plasma membrane, it is universal in nature. Unlike that of a cell wall, we all know there is another protective layer, that is the cell wall. We have discussed before. Both of the structures, they mainly help in giving protection, and also they will be holding the shape of the cell. Okay, but the cell wall, it is absent in the case of certain organisms, but the plasma membrane, it is present in all the living organisms, and hence we call it as universal structure. So unlike that of the cell wall, I have told you this is universal. Then what is the major differences between the cell wall and the plasma membrane? Generally, we say that the cell wall is a dead entity, and the plasma membrane is said to be a living entity. What is the major difference? How do you distinguish this? Both are protective. As I said you before, both are protective, and also they will mainly hold the shape of the cell. They have the similar functions. Then why the cell wall is said to be the dead entity and the plasma membrane is called as a living structure? What is the major difference? So let us see. So why the cell wall it is called as a dead entity, children? The cell wall it is called as dead entity because the living reactions are very few. If you take the case of the plasma membrane it is directly in contact with the cytoplasm, and it will show versatile functions. Many functions are carried on this thin membrane, and it is directly in contact with the living cytoplasm. Many biomolecules, many proteins, and different type of structures will be present on this plasma membrane, and which will show numerous functions. But the cell wall, it will be having very few functions. Okay, so let us see the differences here. One, I have told you that metabolic reactions will be less. So with respect to cell wall, I am writing this point, children. Metabolic reactions will be very less in the case of cell wall. Then what about the plasma membrane? Many metabolic reactions are continuously taking place over this thin layer of plasma membrane. And second important thing, it is very stiff and it is continuously changing. So over a period of time, you can see many changes that are taking place in the cell wall. Different type of depositions are being laid on this cell wall. Okay, and what about the plasma membrane? Plasma membrane, it is constant, its structure is constant. Okay, it is not at all stiff, it is very flexible structure. Okay, so plasma membrane, it is flexible in nature. And what is the third important thing? The cell wall, it is permeable in nature. It will allow almost all type of molecules inside the cell. But what about the plasma membrane? The most important feature of the plasma membrane, it is acting like a barrier. Why we are calling it as a barrier, children? Why? Because it will be allowing selective molecules to enter inside the cell. It will be allowing small non-polar molecules easily inside the cell, but for remaining molecule, major category of molecules, they will be obstructed. The membrane will be acting like a barrier to these molecules. Okay, so this is an important thing. The cell wall is permeable in nature, whereas the plasma membrane, it is highly selective in nature. It is said to be selectively permeable in nature. Alright. So this is permeable and this is selectively permeable in nature. Okay, it will allow almost all the substances inside it. And with the increase in age of the plant cell, then the case will be different. If lignin is deposited or different type of substances are present, in that case the cell wall will not allow any substance to pass into it. Otherwise the cell wall will be allowing almost all the molecules to pass into the cell. But the plasma membrane will be having a different, it will be unique. Why? Because it is selectively permeable. It will allow only few substances. What are the few substances? The simple, you can say the gases, or you can say the water molecules and the non-polar molecules. They can pass or traverse this membrane very easily. But what about the polar molecules, children? We all know that the nature of plasma membrane, it is non-polar. So as it is non-polar, it will allow only non-polar substances easily, and it will be obstructing the entry of the polar substances. Okay, so that we'll be discussing in detail later. So these are the important points, or the differences between the cell wall and the plasma membrane. Okay, so overall, if you see the plasma membrane, how you will define the plasma membrane? Plasma membrane, it is ultra thin, elastic, flexible, and it is dynamic in its nature. So it is very dynamic. It will have numerous functions, and it will be selectively permeable. The most important point here is the selectively permeable. Its permeability, the selective nature, it is very, very important. Not only that, it will be acting like a barrier. It will separate the cell exterior and the cell interior. We have different conditions. The cell environment, it is constantly maintained over the cell. Okay, so inside the cell we have different set of conditions, and these are separated from the cell exterior, which have other set of conditions. So how these conditions are separated? With the help of this particular structure, which is the plasma membrane. Okay, so these are the most important functions of the plasma membrane. Okay, so this is regarding the plasma membrane. So here I have given points, children, you can note it down. And next, this plasma membrane, it is known with different names. Okay, generally there are other terms which are given to the plasma membrane. Let us see the terms here. Plasma membrane, it is also known as cell membrane. What it is known as, children? Cell membrane. So this is cell membrane. It is also called as plasma lemma, and we have another term that is used, that is cytoplasmic membrane. Okay, so we have different names. So we have different names that are used. These are synonyms for the plasma membrane, children. Generally, the cell membrane and plasma membrane, these are mainly used. In certain books, the cell membrane is the word used for the external protective layer which is present above the cell. So generally we have the organelles inside the cytoplasm, and there even we have this plasma membrane, right? So the plasma membrane which is present inside the cell, it is generally said to be the plasma membrane or a cytoplasmic membrane. But the cell membrane is a term, it is used for the membrane which is completely covering the cytoplasm. So which is present externally, we call it as the cell membrane. In certain books these terms are used, children. in a specific way, but it is not required. You can use any term here. Cytoplasmic membrane, plasma membrane, cell membrane and plasma lemma all are same. Different scientists has given names. We are not discussing with the names given by the scientists. It is not important here. And next, coming to the structure of the plasma membrane. So before discussing with the structure of the plasma membrane, we will see the chemical composition of the plasma membrane. So let us quickly look into the chemical composition here. The two important structures which are forming this plasma membrane, you have to remember this. One is lipids and second are the proteins. The majority of the structure which are almost equal in composition are the lipids and the proteins. The quantity of the lipids here, it is ranging between 20 to 79%. Massive amount of lipids are present along with the proteins, and the proteins will be having almost the similar composition, that is around 20 to 70%. And next is of water. Water is 20%, and next are the carbohydrates. It is around 1 to 5%. Along with all these biomolecules, we have enzymes. So this is a very thin membrane and lot of metabolism is taking place over this thin layer. And all these events, metabolic reactions, are assisted with different type of enzymes. We have closely 30 type of enzymes are present on the plasma membrane. So all these are the biomolecules which are present or which are forming this complex structure, that is the plasma membrane. Okay, so this is chemical composition. Now let us see the discovery of the plasma membrane. Lot of theories has put forward to prove the existence of the plasma membrane. Why? Because it is very difficult for a investigator to isolate a plasma membrane, like the structure which is very delicate, very fragile, right? So this is very thin, ultra thin structure. So it is very difficult for a person to isolate and study this compound. Okay, and hence many investigators has worked on this plasma membrane. We are mainly discussing with the three important models of the plasma membrane along with the works of different type of scientists I will be discussing now. Okay, so let us see. Here are the models I have given, children. So for the first time, the existence of the plasma membrane, it was discussed, or you can say the existence of the plasma membrane was suggested by the scientist Overton. Overton in 1895, what he has observed? He has observed that the plasma membrane, it is easily soluble in the fat solvent. What is fat solvent, children? Fats are generally dissolved in the non-polar solvents. Non-polar solvents like formaldehyde, we have acetone and chloroform. All these non-polar solvents, these lipids are generally dissolved inside the non-polar solvents. So this was the first thing which was observed by the Overton. And later he has observed that this plasma membrane, it is easily allowing the entry of non-polar molecules. So what is the second observation? One, he has observed that this is easily dissolved in the fat solvent, that is non-polar solvent. And second thing what he has observed, children, he has observed that the non-polar molecules are easily passing through this membrane. And what he has suggested finally, he said that maybe this plasma membrane, it is made up of fat-like substance, that are lipids. Okay, so the first postulate given by the Overton in 1895. So he told that the plasma membrane, it is made up of lipids. Okay, so these are the structures. This is the structure of the plasma membrane, means these are the lipid molecules. Okay, all these are lipids. We don't have any proteins here. Simply they have discovered that lipids may be present. And next we have the scientist Hober and Frick. In 1910 Hober and 1925 Frick. Both of them coincidentally, both of them has observed that this plasma membrane, it is having very low electrical conductivity. Electrical conductivity, it is very low. Why? Because the plasma membrane, it is generally made up of lipids, and these lipids are non-ionic species. As they doesn't occur in the ionic state, they will be allowing less charge to be passed inside it. So this was an observation made by Hoover and Frey. They said that there is low electrical conductivity on this plasma membrane, and hence they have convinced that the plasma membrane, maybe it is made up of the lipid molecules. Okay, low electrical conductivity. Why? Because lipids are present here. We all know these lipids are acting like insulators. These are not conductors, children, these are insulators. Now you can understand that how this external environment and internal environment are maintained. This is acting like a insulator, so it is present inside, and outside we have saline environment. Yes, we all know the cell exterior or interior, it is just like saline. It will be having different type of ions, minerals which are present inside and outside as well. So these are separated with the help of the plasma membrane. And after that we have Gorter and Grindel. So in 1925 and 26, Gorter and Grindel, they have studied and they have extracted the erythrocyte membrane. So here we can't isolate this membrane intact. Why? Because I have told you it is very difficult to isolate such a thin membrane. We can simply extract the molecules that are present on this membrane by dissolving in certain solvent, and we can study it. Okay. So in those days, now we have different techniques, children. We can isolate this membrane. So then the Gorter and Grindel, they have extracted the erythrocyte, the RBC membrane, and what they have observed, children, the RBC membrane was extracted in the solution. It is spread all over a molecular film. Okay, so it is spread over a molecular film. Then what is molecular film here? In order to obtain these biomolecules, in order to study these biomolecules, we will be using certain film-like structures in the laboratories. And these films are called as molecular films. And this molecular film, it is having affinity for the biomolecule to be attached to it. The lipids are the major biomolecules of interest. And here we have the tape or molecular film, which will be having the molecules which will have affinity to attach to these lipid molecules. So such films are made artificially in the laboratories, and later these biomolecules will be placed into the solutions along with these molecular films. Then what will happen? All these molecules will slowly attach onto this molecular film, and they will be completely stabilized on this film. So that it is very easy to take this molecular film under microscope and you can observe it easily. Okay. So this is how the structures are being studied at that time. And these molecular films are just like adhesive tape or glue-like substance onto which these molecules will attach or adhere. Okay. So this is how they have studied the RBC membrane, and what they have observed. Normally, the predicted thickness of this membrane was said to be 3 to 4 nanometer. When they have observed directly under the microscope, they observed that the plasma membrane is having 3 to 4 nanometer thickness. But when they have dropped into the solution, the thickness will be very less, and also the length of the membrane, the amount of the substance, it is drastically increased. They will be having certain prediction that the membrane thickness, if it is 4 nanometer, it should extend till certain limit. But it has exceeded the limit and it has completely covered the molecular film. Then what is the reason? Then they have assumed that these lipid molecules are present in bilayer, and hence when they have dropped in the solution, they have to, let us say, they have to spread till 5 centimeters. But now what happened? When they dropped in solution, it has spread to 10 centimeters, completely covered the molecular film. It means the lipid molecules are present in a bilayer. So this was their assumption. Okay. So now Overton and Hoover, they have mainly discussed the properties of the plasma membrane, and Gorter and Grindel, they have given an important postulate, that is the lipid bilayer. For the first time, the lipids are present in 2 layers was given by the scientists Gorter and Grindel. Okay. So after this, the major discovery, the major breakthrough for the plasma membrane, it was received in the year 1935 with the invent of the sandwich model, that is given by the scientists Davison and Danieli. So Davison and Danieli, what they have proposed here? The Davison and Danieli, yes, they have studied these erythrocyte membranes, the RBC membranes again, and they have given a model called as sandwich model, or it is also known as a PLP model. There are three names given to this model. It is known as sandwich model or PLP model, or it is also called as trilamellar model. Okay, so different names are given here: sandwich model, trilamellar model, or PLP model. It is a name given for the model proposed by the scientists Davison and Danieli. And here I have missed a point here, children. Gorter and Grendel, they have also explained that this lipid molecules, they are arranged in such a way that they have two regions. What are the two regions? Hydrophobic and hydrophilic region. This hydrophobic region, according to Gorter and Grendel, the hydrophobic region is in the center. Okay, so it is like this, center towards the inner side, and the hydrophilic region is facing upwards. So this is said to be the head region, and this is the tail region. So according to Gorter and Grendel, yes, they have also studied the structure of this lipid. The lipid will have two parts, head region, tail region. And this head region is said to be hydrophilic in nature. And this tail region is said to be hydrophobic in nature. What is hydrophilic? Hydrophilic is water loving. So it will attract the water molecules. And the tail region, it is hydrophobic, which are mainly, these are the fatty acid tails, which will be hitting hydrocarbon tails, and they will be hitting this water. Okay, and hence they are presented inside, and the head region, which is hydrophilic, they will be facing outside. Okay, so this was a structure given by the Gorter and Grendel at that time. And next we have this PLP model, trilamellar or sandwich model. According to Davison and Danieli, here the lipids are arranged in the bilayer. So this is the bilayer, and this will be called as L, lipid layer. And we have two other layers, which are the protein layers, which are present above and the below of this lipid molecule. So lipids are sandwiched in between these two protein layers, and hence this is known as the sandwich model. As they have three layers, 1, 2 and 3. So this was a discovery of the Davison and Danieli. They mainly studied the surface tension of the molecules here. The lipids, when they have observed in the solution, these lipids are solubilized or present in the solution. The lipids are shrinking to its minimum size along with these proteins. So when they have studied the surface tension, you all know that surface tension is nothing but the molecules, they will attain or they will shrink into its minimum size because of the surface tension. And here, to shrink, so proteins are absorbed, and hence the lipids are shrinked into its minimum size. This was also given by the scientists Davison and Danieli. According to them, these proteins here, if you observe, this is one protein layer, this is another protein layer, and these are the lipid molecules arranged in two layers. And what is this gap I have given, children? This gap is nothing but a channel here. Okay, it's a protein channel made. And at that time, they have observed that all the proteins which are present will be of different nature. Okay, the proteins will be having different type of structures. They have alpha helix, beta sheet, that we'll be discussing in zoology again. So all these type of proteins were observed by the Davison and Danieli. So these are the most important postulates given by Davison and Danieli in 1935. And next we have the Robertson. So after Overton, if you observe the evolution of the structure of the plasma membrane or progression, it has started in the year 1895, okay, with the invention of the lipid molecules by Overton. At that time, in 1895 till 1925, it was known that the lipid molecules are mainly present in this plasma membrane. And later, with the discovery of Davison and Danelli in 1935, it was known that the proteins as well as the lipids both are present in the structure, both are present in the plasma membrane forming this structure, okay. And not only that, children, here in the sandwich model, they have explained regarding the type of lipid molecules. The lipid molecules which are present, they will be having hydrophobic and hydrophilic region, and their arrangement was explained, which is very similar to the assumptions given by the Gotter and Grendel. They have explained regarding head region and tail region, hydrophilic, hydrophobic, everything was same, but simply the most important thing you have to remember, they have clearly explained the type of lipid present in the plasma membrane. So the majority of lipids present in the plasma membrane are said to be the phospholipids. That was given by Davison and Danelli, okay. So two important things I have told you. One, they have discussed with the asymmetry of the protein, different type of proteins on the membrane, different type of proteins are present, was discussed by the Davison and Danelli. And second thing is the lipid molecule, the lipids, the majority of the lipids, which are different type of lipids will be present, but majority of the lipids, the major lipids present in the plasma membrane are the phospholipids. So these are the two important things that were given by the Davison and Danelli, okay. So these are the important points, okay. PLP model, trilamellar model, and protein sandwiched. The lipids are sandwiched between the protein layers. 1, 2, 3, we have 3 layers, hence it is trilamellar model, protein, lipid and protein model, okay. And what are the two important things I have told you? They mainly studied the surface tension of the lipid molecule. The lipid molecules, they have shrink to its minimum size with the absorption of these proteins, okay. And hence they have told that the proteins are helping in the shrinkage of these lipid molecules. And another thing, the phospholipids are generally present in majority in the plasma membrane. And the last thing which I have told you is the asymmetry of the protein. The proteins are of different shapes, not similar. They all are dissimilar in nature and they have different functions. It was given by the sandwich model, that was which was given by Davison and Danelli. Okay, and next coming to the model given by Robertson. Robertson has given the unit membrane model in the year 1959. This is nothing but a slight modification of the sandwich model. Okay, so sandwich model, it was redefined by the Robertson and he has given certain postulates. Let us see the postulates of the unit membrane model. The first thing which he has explained is regarding the structure. The structure of the plasma membrane, according to Robertson, it is universal in nature. It is universal and it is present for all type of cells. This was given by scientist Robertson. Okay, and hence he called this plasma membrane as a unit membrane. Unit membrane, why? Because it is present over all the cells as well as the cell organelles and it is similar in nature, and he called it as a unit membrane. Okay, even the organelles will be having this plasma membrane. It may be 1 or 2, the structure will be same. If you take the case of mitochondria and chloroplast, these organelles will be having 2 membranes. Okay, the plasma membrane, it will be of 2 layers there, but the structure will be the same. Each layer will be resembling this particular structure that was given by the scientist Robertson. So this is universal unit membrane and it is present over all the cells along with the cell organelles. This was given by scientist Robertson. And second important postulate. So next important thing, when he has observed under electron microscope, he observed that there are 2 layers. According to him, there are 2 layers. What are the 2 layers? One is electron dense layer and one is the electron transparent layer. Here, the electron dense layer are the protein layers and electron transparent layers are the lipid layers. Okay, so this region, it is said to be electron transparent. So the structure, it is mainly observed under electron microscope, not the light microscope. In light microscope, we use light for focusing on the particular structures. Here, electron microscope will use electrons. Okay, so electron microscope, when he has observed the electron transparent region, it is the lipid region, and the electron dense region, thick region, it is the protein region. And next important thing regarding thickness of the membrane. Here the thickness of the membrane, the overall membrane will be having 75 angstroms, was given by Robertson. 75 angstroms, which is 7.5 nanometer. Okay, 1 angstrom equal to, here I will write, so 1 angstrom equal to 0.1 nanometer. In nanometers, if you want, it is 7.5 nanometer. Okay, so according to him, 75 angstroms is the thickness of the plasma membrane in total, and again individually each protein layer will be 20 angstrom, that is 2 nanometer approximately, and the middle central lipid region, it is around 35 angstroms. In total, we have the 75 angstroms. So this was the thickness, predicted thickness given by the scientist Robertson. Then why these models has failed? Where they have failed? Mainly, here they could not explain three major important points. So the first important limitation or the failure in these models, that is unit membrane model and the sandwich model, they could not explain the movement of polar molecules inside the cells. Yes, they have explained that the non-polar molecules can easily pass through these membranes. Yes, non-polar can pass because the lipids are non-polar and they can pass. What about the polar molecules? That was not explained by the scientists Davison, Danelli and Robertson. And second important limitation or failure is the thickness. The predicted thickness according to Robertson, it is around 75 angstrom, that is 7.5 nanometer, but there are cases where 8 to 10 nanometer thickness was also observed. So that was an exception for their postulate. So what is the next important thing? They could not explain the membrane fluidity. That is, according to them, the membrane is very static. It will not show any movements, but we all know that the membrane is flexible. It is very fluid in nature. Right, so it will be having relaxation, contraction, different type of forms and shapes can be attained by this plasma membrane, which was not explained by the two models. One is sandwich and the unit membrane model. So these are the three major failures or limitations of the sandwich model as well as the unit membrane model. So these are the three important limitations or the three important failures of these models where they could not explain. That is mainly the properties of the membrane. And hence these are said to be the failure models. And next comes the most important model of the plasma membrane, that is fluid mosaic model. These are rejected models. So these are the major failures or limitations of these two models. Okay, and hence these are the rejected models. After these models, there are many scientists, they have explained regarding the proteins, the nature of proteins. The proteins are mainly said to be globular, but all those are the failure models. And the most accepted model till date is the fluid mosaic model, and this was given by scientists Singer and Nicholson in the year 1972, and we'll be discussing in detail with the fluid mosaic model now. Okay, so this is regarding the progression of the or evolution of the structure of the plasma membrane. Though it is very delicate, it took a lot of time to the investigators to investigate the properties as well as the structure of the plasma membrane, and finally there is a successful model that was given by Singer and Nicholson in the year 1972. Okay, so we'll be discussing in detail regarding the fluid mosaic model.